The first thing to do upon receipt of any sample is to assign the sample a specific lab number. In our lab, we use a 6-digit number. The first 2 digits represent the year, and the remaining 4 digits ascend with each sample. This sample number, along with a patient name, is then put on each sample, whether it's a slide or a cell line, and it's also put on the score sheet and then in the notebook. In the lab notebook, you can also record the type of sample, the testing that you will do. In your case, it'll generally be T814 and MIC, along with their hybridization date, and eventually your results. The processing of a touch prep or FNA smear slide is very simple. We'll basically just be taking our slide and dipping it into fixative, which is 3:1 methanol to acetic acid, for 5 minutes. This will fix the cells onto the slide for further processing. After 5 minutes, you can remove the slide. Then you can either air dry or blow dry. After that, the slide is ready for further processing. The process of a cell line requires a few more steps. First, we will decant our cells into a centrifuge tube. The centrifuge tube will then be spun at 1,000 RPMs for 8 minutes to give you a pellet. Once you have this pellet, you will remove the supernatant, agitate the pellet, and add 3:1 methanol to acetic acid. Now, the amount you add will depend upon the size of the pellet, but generally 3 to 6 mils should be good. At that time, you will spin the centrifuge tube again, and again it's at 1,000 RPM for 8 minutes. With the pellet that you receive at the end should be clean, fixed cells. These cells we will now be dropping onto a slide. So the first thing I want to do is remove the fix that is on the pellet. Again, we will agitate our pellet to mix it up. And at this point, I want to add fixative until the solution is still turbid with cells, but you can see your glove through the solution. So after adding a little bit of fix, I'll mix it up and check. So the solution's still cloudy, but I can just see my glove through. This should be a good density of cells for dropping. Now I'll take a slide. So we'll now drop 2 drops, one for each probe that you are going to be adding to the slide later, the 814 and the MIC. After dropping, you'll put on the 37-degree hot plate for 1 minute or less. It's basically just to dry off the excess liquid. Once the slide is dry, you'll want to label it right away. The important things to put on here will be the patient number, the patient name, and the probes that you're applying. The slide is now ready for further processing. Our cell line slides are now ready to be processed through the Copeland jars before hybridization. Our first step is 10 minutes in 2x SSC. Just dunk it in there, wait 10 minutes. I'm not going to. We will then move into 70% ethanol at room temperature for 2 minutes. And these alcohols will escalate from 70% to 85% to 100%, and they are all for 2-minute periods. After that, we'll be ready to move on to the denaturing step, which is 70% formamide at 73 degrees. That step will last for 4 minutes. From there, we go into a series of cold alcohols. We'll now run through the cold alcohols for 2 minutes apiece. These are at minus 20 degrees. Again, they go from 70% to 85% to 100% ethanol. Then after the last 2 minutes, you're ready to take your slides out, and they can either air dry or blow dry. I usually blow dry them. The slide can then be placed on a 37-degree hot plate and is ready to be hybridized. The processing of touch preps and smears requires a few additional steps, but again, we'll start in 2XSSC for 10 minutes. After that is a somewhat important step where we move into pepsin. So pepsin will help to digest extra proteins that may interfere with hybridization. And they will be in there for 5 minutes. That is also at 37 degrees. After that, we will move on to PBS. PBS is at room temp. We'll do that for 5 minutes. Before moving on to formaldehyde for 5 minutes. After that, we have one more PBS step for 5 minutes. This process will then proceed just like the cell lines. We'll move into room temp alcohols for 2 minutes apiece. Moving from the 70% to the 85% to the 100%, each for 2 minutes, before finally moving into the formamide for 4 minutes. Again, this is still at 73 degrees. From there, we'll move on to the cold alcohols. We'll continue on along the cold alcohols, again from 70% ethanol to 85% ethanol to 100% ethanol, each of these for 2 minutes. When you're done with the final 100% ethanol, you can air dry your slide or blow dry it. The slide can now be placed on the hot plate, the 37-degree hot plate, and is ready for hybridization. Before we can hybridize, we need to make our probe. So the probe comes from Visus as a concentrated probe, and we need to dilute it down. So we'll grab 5 microliters of the Visus stock solution. Place it in our empty Eppendorf tube. Next, we'll need 10 microliters of sterile water. So you can readjust your pipette or just get 5 microliters twice. And finally, we'll need 35 microliters of LSI buffer. This buffer is kind of viscous, so it's nice to leave it out at room temp for a little bit before using. Now I will want to briefly spin down, and then I will denature it at 73 degrees by floating it in the water bath for 5 minutes. One last spin and the probe will be ready to use. The final step of day 1 is actually adding your probe. I add 3 microliters of each probe we're going to use. I'll start with 814. As my slide indicates, the 814 will be on the left and the mic will be on the right. So 3 microliters on the left. Now I'll add mic, again 3 microliters. Finally, we'll cover with these 12mm circle cover glass. You may want to use tweezers to get these cover slips. I've been doing it too long to do that, I guess. Then we will add rubber cement and put them in the humidity chamber. If there are any large air bubbles, you can try to push them out. I don't see any here, and generally it's not worth messing with, but if they're extra large, you can try pushing on the cover slip. place it in the humidity chamber. The humidity chamber already had liquid added. It's just DI water. I fill it up and then I dump off the excess, and that should keep it moist overnight. From this point, it'll go into a 37-degree incubator overnight before being washed in the morning.